Hi everyone, this is going to be a simple and concise video on T cell differentiation. So, we know that the bone marrow forms both the B cell and the T cell. So, from the bone marrow, I have the T cell precursor forming. And the thing is, this is going to be double positive. That means it is going to have both CD4 and CD8. After it forms in the bone marrow, it comes to the thymus for maturation. And here something very interesting happens. So we have our CD4 and CD8 positive T cell. So in the cortex of the thymus, there's going to be epithelial cells. And if this T cell binds with affinity to MHC1 epithelial cells, it is going to differentiate into CD8 T cell. The CD4 part of it is going to be downregulated. If it binds with affinity to the MHC2 epithelial cell, this is going to become a CD4 cell. And the CD8 part of it is going to be downregulated. And if there's a T cell that doesn't bind to any of the self MHCs, it has no affinity to the self MHCs, then it is going to undergo apoptosis. If there is no affinity to self MHCs, it is going to undergo apoptosis. And this process, where this is going from a double positive cell and it is becoming CD4 or CD8 and the others are being eliminated, this is called positive selection. And we have seen that positive selection happens in the cortex of the thymus. So now from the cortex of the thymus, our cells are going to journey into the medulla. And in the medulla, what happens is, if these cells interact with self-antigens, that is the antigens that are expressed on the cells that exist in our body, they will be eliminated. Because if our T cells are going to be attacking our own body, then that is going to lead to autoimmunity, and we don't want that. So, the T cells that interact with self-antigens undergo apoptosis. And this is called negative selection. And negative selection happens in the medulla. Now, from here, they are going to go into the lymph node. And the thing is, the CD8 T cells are, ready to go whenever from now. But the CD4 T cells have a little more of differentiation to undergo. They have to take on more specific roles. And these CD4 or helper T cells will differentiate into specialized forms on the basis of the interleukins or the substances that they are exposed to. The things that they encounter will lead to the specialization of the CD4 helper T cells. To make it clear, if the CD4 helper T cells comes into contact with interferon gamma or interleukin 12, it is going to differentiate into a Th1 cell. If the CD4 helper T cell comes into contact with interleukin 2 or interleukin 4, it is going to differentiate into Th2. If it comes into contact with TGF beta or IL interleukin 1, interleukin 6, then this is going to differentiate into Th17. Then lastly, if it is exposed to TGF beta or IL interleukin 2 again, it is going to become a regulatory T cell. Now, what are the functions of these T cells? Why are they specializing into these subsets? The Th1 cell is going to release interferon gamma and interleukin 12. If you recall, these play a very important role in the pathogenesis of tuberculosis. Now, we have a macrophage and we have the serpentine factor of the tuberculosis helping it to invade complete phagocytosis. So this phagocyte is going to, with the TB bacteria inside, it is going to present this to a T cell, to a CD4 T cell. Now, it also releases interleukin 12. So this is going to help T cell differentiate into Th1 subset. and this will release more interferon gamma and interleukin 12, and this activates the macrophages, and we have the formation of a granuloma. And to be specific, there's a caseating granuloma in TB. This was just a quick detour because I wanted to revise the TB part of it, because it arises everywhere, every question, every subject. There's going to be tuberculosis in medicine. So now, what do Th2 cells do? They are going to recruit, they are going to lead to the release of interleukin 4, 5, 6, 10, and 13. So again, to recall, interleukin 4 and 5, these play an important role in hypersensitivity reaction 1, atopy, anaphylaxis. And they help recruit eosinophils, and there's an increase in release of IgE antibodies. And then, what do Th17 cells do? These are going to release interleukin 17, 21, and 22, and these will recruit neutrophils. Oh, another thing that I wanted to mention here is there's recruitment of CD8 also that takes place here. And then lastly, regulatory T cells. And these are super important because if we don't have anything controlling the immune attack that happens, then there's going to be more destruction than the healing that it initially started out to make happen, right? So regulatory T cells will lead to the release of TGF beta, interleukin 10, and interleukin 35. And these are anti-autoimmunity. That means they basically maintain tolerance. Now, this was about the CD4 T cells. A quick thing about the CD8 T cells is that they are going to release something called perforins and granzymes, which are preformed proteins, and these come packed in these cytotoxic granules. And these are going to help kill virus-infected cells, neoplastic cells, and also donor cells, donor graft cells. And this happens by inducing apoptosis. So that was hopefully a very simple, very concise, but detailed review of T cell differentiation, and I hope it helped. Thank you for watching.